Hello guys, and welcome back to the Doctor's Medicine channel. I am yours Chiremba Englas, and today we'll be doing the histology of the liver and the gallbladder. Okay, so to begin with, we have the basic functions of the liver. Of course, the liver has more than 500 functions, but I'll just list the basic functions here. So first of all, the liver is an endocrine gland. Why? Because it synthesizes and it secretes plasma proteins, and also it secretes hormones into the bloodstream. Alright. It also is an exocrine portion where we focus mainly on the synthesis and the secretion of bile into the biliary duct system. Right. It also has a metabolic function in metabolism of carbohydrates and fats. So you have storage function, glycogen, lipids, fat-soluble vitamins, iron. It also acts to filter the blood, removing worn out and damaged red blood cells, also removing microorganisms. The liver also has a function in detoxification of toxins, drugs, and harmful substances. So, in the fetus, the main or the primary function of the liver is to produce or to form blood cells, which is a hematopoietic function. So, for the general histological structure, we have our liver. Like any other organ, it has parenchyma and stroma, where we say the parenchyma is the functional tissue and the stroma is the supportive tissue. So, the parenchyma of the liver is derived from the endoderm of the gut tube. Alright, and we say that the hepatocytes are the main cells in the parenchyma of the liver, and these hepatocytes are actually epithelial. Okay. Then the stroma of the liver is derived from the mesoderm. So this includes the connective tissue, the blood vessels, reticular fibers, and the connective tissue capsule, which we refer to as the Gleason's capsule, covering and surrounding the outermost part of the liver. Right. So this Gleason's capsule, as it approaches the region of the portal triad, it actually thickens. Okay. We also have connective tissue trabeculae extending into the liver, right? And this is an effect of partitioning or sort of like forming interlobar septors. But however, these interlobar septors, they're mainly visible in the liver of animals like pigs, but in humans, interlobar septors are not that visible. Okay, all right. So for hepatocytes and sinusoids, we will look at them in depth in the following slides. Okay. So as you know, for the blood supply of the liver is pure blood supply. We have blood supply from the hepatic artery. We have blood supply from the portal vein. So the hepatic artery brings us oxygenated blood to the liver, and the portal vein brings us deoxygenated blood, which is a higher nutrient content. Okay, so this hepatic artery and this portal vein, they will form smaller branches, okay, which will go into the parenchyma of the liver, and these now we term them as the hepatic arterioles and the portal venules, right? They will run alongside the peripheral areas of the hepatic lobule in what we call the portal tract alongside with the bile duct tubes. Okay. So as you can see here, this is the functional unit of the liver. We'll define it in the next slide, but as you can see, it's a hexagonal structure. Okay. And then at the periphery here, we will find mainly three structures here, including the hepatic arteriole here, including the portal venule and the bile duct tube. All right. So this blood now, the hepatic blood from the hepatic artery and venous blood from the portal vein, they will mix in the liver sinusoids. All right. They will mix in these spaces, and then they will later drain in what you call the central vein. Okay. So this central vein will drain into what you call sublobular veins, because remember, the central vein can also be termed centrilobular veins. Okay, so the centrilobular veins, they will drain into what you call the sublobular veins. So if you can see this picture here, these are centrilobular veins here at the center of our lobules, and they'll unite and drain into what you call a sublobular vein, and this sublobular vein will actually join up with other sublobular veins and form the hepatic vein. Okay, so for the structural unit of the liver, we have what you call the hepatic lobule. All right, so this hepatic lobule is a structure in which the liver parenchyma is organized into a hexagonal structure with a central vein at the center, and peripherally at each of the 6 corners there is a portal area, right? And then within this portal area we find the portal triad, we find nerves, we find lymphatics, and we find fibrous connective tissue. Recall that the portal triad is comprised of the hepatic arteria, the portal vein, and the bile duct too. All right, so this is the general structure of the structural unit of the liver, which we term the classic hepatic lobule. So what happens is that this here is our central vein here, right? This one, our central veins. So from around the central veins, cords of one cell layer thick plates will radiate from the central vein, and these plates, we actually term them as the hepatic plates or hepatic cords. All right, so in between the hepatic plates in this region, we have what we call the hepatic sinusoids. Right, so these sinusoids are sort of like dilated capillaries. Okay, they have a lining of endothelial cells, although in the liver this lining of endothelial cells, it is fenestrated to allow rapid movement of substances across from the hepatic sinusoid into the hepatocytes or in the reverse direction. Okay, so it has an endothelial cell lining, a fenestrated endothelial cell lining, and these endothelial cells have a discontinuous basal membrane. All right, so this will facilitate the rapid transport. Also note that within the sinusoids we find blood. Remember, I said that blood from the hepatic arterial and the portal vein, it will mix within the sinusoid. Okay, so this blood is in direct contact with the hepatocytes. This increases the rate of transportation of substances across or from one region to another. Okay, so within the sinusoids, we will find that there are also macrophages. So we find certain types of cells which we actually call the Kupffer cells. These are resident macrophages which will remain in these sinusoids, okay, so that they'll phagocytize any microorganisms which will invade the liver, or they'll also phagocytize worn out red blood cells. So our portal tract, remember, this one is our portal tract, okay, which contains the portal triad. So blood from the portal tract or portal triad, it will pass into the hepatic sinusoids first in here. So from the hepatic sinusoids, it will continue along these ones, and it will reach the central vein. Or remember, I'll say it's a central vein or a centrilobular vein, and from the centrilobular vein, it will drain into the hepatic veins, okay, via the sublobular veins. All right. So for the portal area, it contains the portal triad. Portal area, it contains the portal triad, right, within the fibrous connective tissue of reticular fibers. Okay, so contents of the portal area. First of all, we have the portal vein. This portal vein, histologically, it can be seen as the largest structure. This one, this is our portal vein, right? See, it's the largest structure present, all right? So this is the terminal branch of the portal vein. All right. Then we can also see that this portal vein is lined by an endothelial cell lining of squamous cells, simple squamous cells. All right. It is an irregular lumen, so that's how we can identify it. The hepatic arteriole here, this is our hepatic arteriole. This one, it is a branch of a terminal branch of the hepatic artery. This one, it can be distinguished from the rest of these structures in that it has more of a regular lumen and it has a distinct tunica media here. Remember, blood vessels, we said arteries are distinguished by a well-developed tunica media, this one. Okay, and this one is also lined by an endothelial cell lining of simple squamous cells. All right. Now, the third structure we find in the portal area is a bile duct tube, which is almost similar to the hepatic arteriole, but however, they are different in that the bile duct tube is lined with an epithelium which is simple cuboidal. Okay, you can see these cells; these are round nuclei. They represent cuboidal epithelium. So within this portal triad, within this portal area, we can also find lymphatic vessels, okay, and also nerve fibers too. But however, the lymphatic vessels cannot be easily distinguished here because they have delicate walls, so normally they are collapsed, but they are found in the region of the portal area. Okay. So now, let's look at what we call the space of dice. Okay. So the space of dice, we can also call it the perisinusoidal space, right? Why is it called the perisinusoidal space? All right. Remember, so consider this is the sinusoid lumen, and these are endothelial cells. You can see this nuclei here. It's a nuclei of endothelial cells, right? But these endothelial cells, remember, they are fenestrated and they have a discontinuous basal lamina to allow rapid transport of substances from the sinusoidal lumen to the hepatocytes. Okay, so from here to the hepatocytes. This rapid transport is facilitated by the discontinuous basal lamina. Okay, so the space of dice is the space between the endothelial cell lining here and the hepatocyte. Okay, this one is what you call the space of dice or the perisinusoidal space, and within this space we mainly find plasma. Okay, we also find hepatic stellate cells. This one is the hepatic stellate cell, and you can see that it is almost the shape of a star, right? Stellate means it is almost a shape of a star, all right? And this one, they can be also called Ito cells, or they can be referred to as adipose cells. Why? Because they have fat within them. So we can also refer them as adipose cells. They also have a function of storing vitamin A. Okay. Within this space of dice, we also find the reticular fibers which support the endothelial cells, these ones. And in the fetal liver, this is where we find the islands of hematopoietic cells. Because remember, the liver is the main hematopoietic organ prenatally. Okay. So this is our hepatocyte here. Then these ones are joined by tight junctions. Alright. The hepatocytes joined by tight junctions. This structure, we refer to it as a biocanaliculus. Alright. For the concept of liver lobules, we have three main concepts. Okay. We have the hepatic acinus, the portal lobule, and the classic lobule, of which the classic liver lobule is the one which we've been dealing with lately, where we said blood from the peripheral portal areas will drain via the hepatic sinusoids into the central vein. Then the portal lobule, this one concerns with the drainage of bile into the interlobular duct. This one, okay. First of all, let's start with the liver acinus of this one is actually based on the flow of blood within hepatic tissue from the portal area here towards the central vein. So it divides the liver parenchyma into three zones. Right? The first zone is the one which is closest to the regions of blood supply, these ones. So zone number one receives more of the oxygenated blood and more of the nutrients than any other zones. Okay. Then zone number three, being the furthest away, it receives less of these nutrients and oxygen. Okay. Zone number two is an intermediate zone, so it gets intermediates of both. All right. So the functions of hepatic cells in these three zones is different. All right, because cells in this region have a completely different function. They get more oxygen, they get more blood, so most of them carry processes which require oxidative metabolism, such as protein synthesis. Okay, cells in the region number three, they are furthest away from the oxygen and the nutrients, so they'll carry also other functions different from those in zone one. Okay, including glycolysis, lipid formation, and drug biotransformations. Zone number two, it remains an intermediate zone. So this is just the liver acinas of Rappaport. On this concept of the liver lobule, we defined the classic liver lobule, classic hepatic lobule. We defined the hepatic acinas of Rappaport. And then for the portal lobule, the portal lobule is simply drainage of bile into the interlobular duct. So this one just says that the bile which drains to the interlobular duct comes from three different lobules. This one being the number one, this one being the second, and then this one being the third. That just are the summary of the portal lobule. So for the cells of the liver, the hepatocytes makes up 80% of the liver cells. All right. So this hepatocyte is an eosinophilic cell with abundant mitochondria, right? It also has smooth endoplasmic reticulum, rough endoplasmic reticulum, and all other components of the cell. All right. So it has a rounded nucleus here, which may present is euchromatic, okay, and it also presents as polyploidy. Right? So about 50% of the hepatocytes, they are binucleated. Okay. So for other cells of the liver, we have the Kupffer cells. This one we say these are resident macrophages, which phagocytize one-out erythrocytes. These are located within the hepatic sinusoids. So in these hepatic sinusoids, this is where we find Kupffer cells here. This is a Kupffer cell within the hepatic sinusoid. Okay. This one is a Kupffer cell. This histological section was made after putting ink drops within the liver cell, and then these Kupffer cells are now phagocytizing the ink droplets. So these black dots there, this is ink, okay, and Kupffer cells can be seen phagocytizing this ink here. So also they have a function of phagocytizing worn out red blood cells and other microorganisms. Okay. Ito cells, these are fat storing cells of mesenchymal origin. Alright. They also secrete reticular fibers. Okay. They store vitamin A. So ito cells can also be referred to as adipose cells, adipocytes of the liver. These ones, they are located within the space of Dice or in the perisinusoidal space. Okay. So for the flow of blood and bile in the liver, right? So let's start with the flow of blood. The flow of blood, remember we said blood from the portal triad, okay? It drains first into the liver sinusoids. So from the liver sinusoids, it goes to the central veins, okay? From the central veins, it goes to the sublobular vein, and from the sublobular vein, it goes to the hepatic vein. From the hepatic vein to the inferior vena cava. So just this is the pathway, okay. And for the flow of bile now, the flow of bile is opposite to the flow of blood. So when blood is coming from the periphery towards the center, bile is coming from the center towards the periphery. Okay, so we have what you call bile canaliculi. All right, bile canaliculi, these are anastomosing channels formed by the plasma membranes of adjacent hepatocytes. Okay, so bile flows opposite to blood flow from the hepatocyte. It goes to the bile canaliculi. Remember, these are between the hepatocytes, and from the bile canaliculi goes to the bile duct 2. The bile duct 2, remember, it is located in the portal tract, and from the bile duct 2 you go to the intrahepatic duct, right? And from there go to the hepatic duct, the right or the left hepatic duct. Okay, so we can see and we can appreciate here that these are hepatocytes. Okay, then adjacent hepatocytes, their plasma membranes will form what you call bile canaliculi. All right, so from this bile canaliculi I'll go to the canal of Hering here. So this is bile flowing, bile canaliculi, canal of Hering, interlobular bile duct. Okay, this is the same diagram representing the same thing, from bile canaliculi to canals of Hering to interlobular bile duct within the portal area. So take note that in the region of the canals of Hering we find progenitor cells. These progenitor cells will act to replace the worn out hepatocytes. Remember, we said that hepatocytes, they have the ability of regeneration. So this is the function of these progenitor cells. Now let's go to the histology of the gallbladder. The gallbladder, its main function is to store and concentrate bile. The gallbladder does not form bile, but it just stores and concentrates it. So its histological structure: the gallbladder has a mucosa, muscularis externa, and the serosa or adventitia. Take note that it does not have the muscularis mucosa and a submucosa, right? So for the muscularis externa, it is typical of the GIT, but however, it cannot be distinguished into either the longitudinal or the circular layer. The epithelium of the gallbladder is simple columnar with microvilli, is as mucosal enfoldings, okay? But however, it does not have, the gallbladder does not have glands in the region of the body and the fundus. They are only present in the region of the neck. Okay, so for the histology of the biliary tree, take note that the epithelium of the gallbladder is the same as the epithelium for the cystic duct and also for the hepatic ducts, right? And also for the bile duct. So the epithelium of the cystic, hepatic, common bile ducts, they're lined with the same simple columnar epithelium. All right, the cells, they can actually be referred to as cholangiocytes. All right, but however, since we say that these common bile ducts, it will drain into the second part of the duodenum via the major duodenal papillae together with the major pancreatic duct or the duct of Weissan. Right. So take note that the epithelium of the biliary tree is different from the epithelium of the pancreatic ducts. Okay, epithelium of the major pancreatic duct, it's stratified columnar, but that of the biliary tree is simple columnar. Okay, so that will conclude the histology of the liver and the gallbladder. Stay tuned for the next video on the histology of the pancreas and the spleen. Thank you.